Okay. So you should remember we covered cellular respiration in our last meeting. So we're going to start with carbohydrate metabolism. That links in quite nicely. So firstly, we're going to start with the digestion and absorption of carbohydrates. So when you're eating carbs, you're eating them in polysaccharide form, okay? But you can't absorb them as these massive polysaccharide chains, okay? We have to convert them into a monosaccharide form, and this is mostly converted to glucose. Okay, so when we start eating our food, it first goes to our mouth and stomach. And these polysaccharide chains, they're hydrolyzed via an enzyme called alpha amylase, which hydrolyzes about 30 to 40% of starch and into maltose and other polymers. Okay, then we travel into the pancreas, okay, and that also secretes a large amount of alpha amylase, but it's even more powerful. So almost all of your polysaccharides are actually hydrolyzed at this point. In the intestine, it does even more work, okay, because sometimes it's not just converted straight into glucose. There's some disaccharides and other smaller polymers, and they need to be just finely hydrolyzed into glucose. Okay, this little bit here at the bottom, so this isn't so vital to remember, but just have to know that it's not always completely converted into glucose. There's sometimes galactose and fructose, and they have very similar metabolism, and you don't really have to know anything about that other than the fact that they also exist. Okay, so we'll keep going. So now I'm going to talk about glycogenesis and glycogenolysis. Okay, so what happens is that sometimes you've eaten a bit too much glucose, okay, and you want to store that glucose for later use. So we go through this reaction called glycogenesis, okay, so that's basically forming glycogen from glucose. Okay, this particularly likes to happen in our liver and muscle cells, okay, and we'll come back to why, but all body cells are capable of storing some glycogen, okay? Now the opposite is glycogenolysis, which is converting glycogen to glucose, okay? So it's sort of the breakdown. Even though it's the reverse of the end product, so glucose to glycogen or glycogen to glucose, it's not the exact reverse of the reaction, so don't fall into the trap of thinking it's the exact reverse, because there's different intermediate molecules that are produced and different enzymes that are used, okay? We'll keep going. Then we have gluconeogenesis, which is the creation of new glucose, okay? Sometimes we don't have glucose being digested from an outside source, okay? But things like our red blood cells, they always need glucose to produce their energy, okay? So sometimes we can convert glucose from other sources, such as amino acids and glycerol. So all these amino acids, these lactates, these lipids, they're converted back to pyruvate, and then this pyruvate is made, makes glucose, okay? An important note is that oxaloacetate is produced during the first step of gluconeogenesis. That's probably the only step in the reaction that you actually need to know about. And we'll talk a bit more about how amino acids are converted into glucose a little bit later when we talk about amino acid metabolism. So this here is a bit of a diagram of glycolysis and gluconeogenesis. So you can see they're quite similar, but up here they have different enzymes and different intermediate molecules like oxaloacetate here. So don't fall into the trap of thinking they're exactly the same thing, just reversed, because they're not. But they do have their similarities. So that's just, you don't have to know this diagram at all. This is just to show that you can go from pyruvate to glucose and back, okay? But you don't have to know it in detail. Next, we're going to talk about the lactic acid cycle. It's also known as the Cori cycle. They're interchangeable names. You'll just see different lecturers using different names for it. This is basically during anaerobic respiration. So sometimes, say you're running away from a cheetah, I don't know, and you need some access to glucose really fast to be able to power all your skeletal muscles, okay? So what happens is that you have glycogen stores, okay? So remember, glycogen is just the storage of glucose, and you have glycogen stores in your muscle tissue here, okay? Now this glycogen gets converted to glucose, which then makes lactate, okay? So you should remember that from the cellular respiration lecture, okay? And that glucose is then used by the cells in the muscle tissue for energy, okay? And that's used pretty quickly. Now, what happens here is that it produces lactate, and lactate isn't very good for us because it lowers the cellular pH, okay, makes it all a bit more acidic, okay? And that lactate, what happens is we need to get rid of it, okay? We don't like it staying in our muscle tissue. So what happens is that lactate gets released into the blood in the form of blood lactate, and it goes all the way back to our liver, okay, where it undergoes gluconeogenesis and converts back to glucose. So remember, gluconeogenesis is just creating glucose from other sources. So it just converts it back, okay? And it requires ATP for this reaction. Then what happens is that the liver doesn't really need this glucose, so it sends it back into the blood and sends it back into storage in the muscle tissue. That's sort of what happens, okay? The next couple of slides just go into detail about this. You can revisit it, but I just explained it just there. Now we have mobilization of carbohydrates. So sometimes you have glycogen and it needs to be broken down. So most of the time you're eating food and you're getting your glucose from your diet, but sometimes you're not. So say in between meals, you might need to release glucose, okay? And this acts as a buffer to maintain optimal blood glucose levels. And this is especially important for the brain, okay? There's also some cells in your body which can only work on glucose, okay? And that's particularly your red blood cells because they don't have any organelles in them and they don't have any mitochondria. So they only undergo anaerobic respiration of glucose. So it's really important to always have a bit of glucose in the blood. Another reason why we might be breaking down glycogen into glucose is how I mentioned before, when you're using a lot of your skeletal muscles for sudden and intense activity, and it can be broken down into glucose, okay? And so do remember here that lactate is produced. Yeah. So there are two main glycogen stores, like I've said earlier, and that's in your liver and the skeletal muscle. Okay, so the liver is sort of king of metabolism, okay, and everything sort of happens in there. So if in doubt, you just know everything happens in the liver, and we'll cover that a bit later as well. And we also have stores in the skeletal muscle, okay, it makes sense because sometimes we need to use more glucose, say in intense activity, okay. Now, important to note is the actual reaction in these two tissues. So in the liver, glycogen gets converted to G6P, or glucose 6-phosphate, and then to glucose, okay? Then this glucose is sent all around the body to various tissues and is used for its processes there, okay? In skeletal muscle, however, glycogen just gets converted to glucose 6-phosphate, okay? And then this glucose 6-phosphate is directly used by the muscle for its energy production, okay? So this is a really important thing to realize. Now, the reason for why it doesn't actually get converted to glucose, its enzymes in the skeletal muscle can directly just use this G6P, and it makes sense because if you need a lot of glucose being made or being used up, you want to get it as quickly as possible. So if it was converted to glycogen, to G6P and to glucose, it just takes longer, okay? And that's time that you don't have when you're running away from something, for example. Okay. So this graph here is a good sort of overview of when we use what type of source of glucose, okay? So exogenous, whenever you see this word, and it might come up a bit later, this means that you're getting it from an outside source, usually from your diet, okay? So you're actually eating food and you're using that glucose directly. Now you can see after a couple hours, after you've had your last meal, you can see here that we have a lot of glycogen being used, okay? Which makes sense because you're not getting glucose from your bloodstream anymore and you still need some glucose being produced pretty much at all times, okay? So it's using your glycogen stores in your liver, mainly your liver, but can also use it from your skeletal muscle. And remember, there's also glycogen stores in all cells, okay? Don't forget that. But you can see here that we have this yellow line going up, and this represents gluconeogenesis, okay? So this is the other form. Now you can see here, gluconeogenesis seems to peak after a couple of days, okay? And the reason for why, remember gluconeogenesis is making glucose from other molecules, for example, lipids and proteins, okay? But you'd rather not be doing that, especially looking at our proteins. You don't want to be degrading other types of tissues to make glucose if you can avoid it, okay? But after a point, we have to use gluconeogenesis because we have no choice, okay? So you don't have to necessarily memorize this graph, but it's just good to understand it. And if you understand it, you know that you've got it, okay? So we'll keep going. And last bit here, we have hormonal control of carbohydrate metabolism. Now, this is a bit of a random slide in one of your lectures, I believe. I think there's literally one slide on it. It's a bit of a, I don't know why we're learning this, but it makes sense, okay? So hormones are just usually proteins. They can be other things as well. Well, we have three important hormones in relation to carbohydrate metabolism. So firstly, we have insulin, which you should probably all have heard of about now. It's related to diabetes. So insulin is released when the glucose concentration is too high in the body. And what happens is that it primes all the cells to take up that glucose and to actually use it in its cellular respiration. Glucagon is the opposite to insulin. It's released when there isn't enough glucose in the body. And it sort of signals to all the cells to make maybe using its glycogen stores or maybe even going through gluconeogenesis. So you can sort of think of glucagon released in starvation, okay? Epinephrine, this is also just a bit random, but you still have to know it. Epinephrine just prepares your muscles, lungs, and heart for a burst of activity. So you might even know that epinephrine is actually what's found in EpiPens. So if you think about giving someone a shot of adrenaline, yeah, you're just going to think about how it makes the muscles have bursts of activity there. Now, you might get an exam question on insulin, for example. Usually they would actually define some in relation to diabetes, okay? So in diabetes, in type 1, your body doesn't actually make enough insulin. In type 2, your cells don't respond to the insulin. So you're not expected to know that, but they might give a bit of background. They might actually explain that in the question and then ask what would happen if your body doesn't respond to insulin, okay? And you just have to figure that out. Okay, so that's sort of an example of what an exam question might look like. Okay, moving on to lipid metabolism. So remember, lipids are made out of fatty acids and a glycerol molecule, okay? So first we're going to talk about fatty acids and their synthesis. So you can get lipids from an exogenous source from the diet, or sometimes you can actually make these lipids from other substances in the body, okay? So first of all, this should be a bit of chemistry revision, hopefully. So you should remember that fatty acids are long hydrocarbon chains that contain a carboxyl group and a methyl group at the other end, okay? They can either be saturated or unsaturated. Remembering unsaturated is when there's double bonds. And you can have monounsaturated or polyunsaturated. This should all be revision, to be honest. Now, sometimes if you need to make these fatty acids from the body, there's this really important reaction down here that you will have to memorize, okay? So we can have acetyl coenzyme A being converted to malonyl coenzyme A, which is then converted to palmitic acid, which creates the fatty acids, okay? This is really important to note, especially the malonyl coenzyme A, okay? That this is an intermediate in this reaction because you might have an exam question on that. Okay, then these fatty acids can then be converted into triacylglycerols or TAGs, and that's how we will be referring to them, phospholipids and different types of sterols. Okay, so we're going to have a quick overview of that. One more thing is these essential fatty acids. So there are some types of fatty acids that can't be synthesized by the body, okay, and you have to take them by the diet. And there's only two of them, okay, so there's linoleic fatty acids, so it's an omega-6, and linolenic, which is an omega-3. And that's all you have to know for this, really. They might ask you which one of these is a essential fatty acid, and you just have to know that these are the two essential fatty acids, okay? Now we're going to go into TAG synthesis. Again, this should sort of be revision from chemistry, to be honest. So remember that they can be synthesized in the liver and in adipose tissue, which is your fatty tissue, I guess. And they can be found from the diet or from other molecules, okay, from acetyl-CoA, for example. So remember that the structure is 1 glycerol and 3 fatty acid chains. So there's a picture here of a tag being produced. Now, all you have to know is that there's slight differences in the first step of synthesis in the liver as compared to the adipose, okay? You don't really need to remember the actual reaction, But if you can just remember the first step differs, you should be set to go, okay? In the liver, once these fatty acids or these tags are made, they're secreted as something called very low density lipoprotein. And that means that they're sent out to other tissues in the body, okay, other cells. But some of these tags are also stored in the liver, okay? And we'll come back to VLDL a little bit later when we talk about lipoproteins in more detail. Now, in adipose tissue, if they're made in the adipose tissue, they're usually stored in the adipose tissue. And later on, they can be released to other cells and just used by the other cells. We're going to talk a little bit about phospholipid and sterol synthesis. So remember that they also have fatty acids in them, and the body still needs to use them. So phospholipids are found in the membrane of cells, and sterols, particularly cholesterol, is found in the body also in membranes, okay? And it helps the rigidity of the membrane, which should be a revision, hopefully. So a phospholipid, its structure is one glycerol, two fatty acids, and a phosphate group modified by an alcohol, okay? So it's synthesized in every cell except red blood cells, because remember, red blood cells, they're kind of weird. They don't have any organelles in them, okay? So they can't really do much. So please remember this reaction here, that there's a choline plus a phosphatidate makes a phospholipid. Same thing here with sterols. They're synthesized in the adrenal cortex, liver, and gonads. This is quite important to recognize. You might be asked a question on it. And they're made from acetyl coenzyme A plus this enzyme called HMG coenzyme A reductase, and that makes cholesterol. And this enzyme here is really, really important to remember, okay, because you might be asked something about it. Now, the synthesis of cholesterol is upregulated by insulin, which sort of makes sense, okay? Because when you have a lot of glucose, so remember insulin is released when there's a lot of glucose. If you have a lot of glucose, you're making a lot of acetyl coenzyme A. So you may as well just make a lot of cholesterol. Now, it's downregulated by glucagon. So remember, glucagon is released when there's not a lot of glucose around. You don't want to waste your precious glucose on making cholesterol, okay? Because that's not as important as the other, you know, providing constant energy to your red blood cells or your other organs, okay? And this high intracellular cholesterol concentration also downregulates cholesterol, so makes less of it. And this is an example of negative feedback, which we talked about in our last PSP presentation. So it makes sense. If you have too much of it, you don't want more being created. We'll keep going. Okay, so next we're going to talk about lipid digestion, okay, and absorption. And I have this diagram. So the previous two slides are just in written form of what I'm about to say. So basically, if you have a lipid coming from the diet, okay, you have this really large lipid droplet, okay? You see our cell membrane here on the intestine, it's too big to actually absorb, okay? So it needs to be broken down into smaller molecules. So what happens is that it mixes with these bile salts from the gallbladder and it makes it into smaller lipids, basically, and they're called micelles, okay? What this means that firstly it's smaller, but it gives it more surface area for these pancreatic lipases to come and break down these molecules. Because in the end, the only way a lipid can cross the cell membrane is by being converted into fatty acids and glycerol, okay, or monoglyceride. And that's the only way, because they're small enough to just go through here. Whereas this lipid is just too big and it won't cross, okay? And so remember, a lipase, A stands for enzyme, and you can kind of guess that this is breaking down these lipid molecules here. Okay, so it crosses the membrane and it goes inside the cell. Okay, see here, they're still in fatty acid in monoglyceride form. Then in the endoplasmic reticulum, it actually synthesizes them together to make tags, okay? So it seems a convoluted way of saying that it goes from a tag, gets broken down into fatty acids, monoglyceride molecules, crosses the membrane, and then is reformed into a tag, okay? Simply because it can't pass through the membrane in its bigger form. So once it's in there, it gets synthesized and gets packaged into this thing called a chylomicron. Okay, and we'll come back in a second as to what a chylomicron actually is. And it gets released from the cell and goes into these lymphatic vessels. Okay, and then it gets transported to all around your body, okay, as needed. So these fats, they need to be transported to other tissues, right? There's two ways of transporting these lipids, as free fatty acids in the blood or as lipoproteins. So these free fatty acids, they're just fatty acids, okay? And they're released from the adipose tissue as needed and are transported in the blood with albumin. And this albumin is really super important to take note of because you might be asked an exam question on that, okay? Because they can't just run around in your blood without any other support, so it needs this albumin to be transported. Alternatively, a different form of transport is through these substances called lipoproteins. So they're made of protein and fat, and they carry cholesterol, phospholipids, and tags through your bloodstream, okay? So basically, a lipoprotein consists of a hydrophobic core and a hydrophilic external coat. Okay, and this makes sense because these lipoproteins, they're traveling through the blood, okay, which is a watery environment. You want it to be able to actually smoothly be transported in that blood. That's why this external coat is hydrophilic, okay, and it's made out of phospholipid monolayer, free cholesterol, and apolipoproteins, okay, and this is important to memorize what is on this external coat, okay, remembering it's hydrophilic. Now, the core of this lipoproteins, remember its purpose is to mainly transport tags, okay? So it's going to keep its precious treasure inside of it, okay? And because these tags, they're lipophilic, okay, because they're lipids, you want the core to be lipophilic or hydrophobic. That's the same thing, okay? Hydrophobic and lipophilic basically mean the same thing. They're just different ways of saying it. And you're going to have to remember that the hydrophobic core includes insoluble lipids, tags, and cholesterol esters. Okay, so that's important to know. This next diagram is just a diagram of what I've just said. Hopefully that makes things a bit clearer. So you have this coat, which is hydrophilic, okay, and this inside layer with all these tags inside, that's hydrophobic. So this here is a really, really good table. It actually came from a previous PSP called PSPuppers. Feel free to check them out. They were around when we were in first year. They have some pretty good notes and slides. So here we have different types of lipoproteins. I really recommend just memorizing this table. You will be miles ahead of everyone else if you can early on memorize this table, okay? It will make your life so much easier. So as mentioned before, we have something called a chylomicron. So remember, that's released straight after a meal. Okay, and then that chylomicron transports those tags to other body tissues. Okay, you can see here the main components inside this chylomicron. It's mainly tags with a little bit of cholesterol. Okay, now we have these things called VLDL, LDL, and HDL. Okay, I'm going to consider them a little bit separately to the chylomicron. So all of these are released between meals, okay? So they're a little bit separate and they're released from the liver, okay? Because that's a store of your fats there as well. And they transport fats to other tissues in between meals. Now, if you go and look at VLDL first, that stands for very low density lipoprotein, okay? It's made in the liver and its main components are tags, okay? Now, if we look at LDL, low density lipoprotein, that's found in the blood, okay, and it mainly consists of cholesterol. Now, these two are actually linked because VLDL later becomes LDL after the VLDL has given off some of its tags, okay, so that's said here. Oh, here, remnant of VLDL after it gives off tags, okay, so VLDL becomes LDL. And then we have, lastly here, HDL, high density lipoprotein. It's also mainly found in the liver, and it's mainly protein, okay? So this is quite different to these ones, which were tags and cholesterol. This is mainly made from protein. What it does, it actually collects cholesterol from your tissues, okay? So this is also called reverse cholesterol transport, and it returns the cholesterol back to the liver for excretion, okay? Now, I'm just going to explain the relationship between these three. So VLDL, its whole purpose is transporting tags from the liver to the tissue, okay? So that's why a lot of it is made out of tags. Once it's given off its tags, it becomes LDL, And so only a little bit of it is tags, but now most of it is made up of cholesterol, okay? And this then transports the cholesterol to other tissues. Now, this isn't really good, and you might have heard of some things like high cholesterol is bad, which is true. You still need some cholesterol in the body, but if you have too much, it can give rise to cardiovascular disease later in life and cause heart attacks and that type of thing. Now, this is where HDL comes in, because this is called good cholesterol, because it returns cholesterol to the liver for excretion. So you want more HDL, which is good cholesterol, and less LDL in your body. Okay? Now, I know this gets confusing with why some things are very low density, why other things are high density. So how I like to think about it is you know how fats float on water? That's because fats are less dense, okay? So if you have a lot of fats, that means they're very, very low density, okay? If we have proteins, and it's all proteins, okay, that is a lot more dense than fats. So that's how you know why they're called very low density or high density. So hopefully that made a bit of sense. And yeah, just memorize this table because you'll be miles ahead of everyone. So these next 3 slides just summarize what I just said, if you want a bit more detail on that. Now, there's 2 main deposits of lipids in the body. So they're found in adipose tissue, which is what you sort of all know as fat. And they're stored in specific cells called adipocytes, okay, which is found in this adipose tissue, okay? And basically they're stored there until these tags are needed elsewhere for energy, okay? And this adipose tissue also provides insulation from the cold, so it's actually quite useful as well. Important to note is that these adipocytes all have lipases. So remember, lipases are the ones that break down these fats. So when they do need to be broken down, they can easily be catalyzed by these lipases and released as fatty acids and glycerol into the rest of the body. Now, the liver also has high amounts of lipid deposits. Remember that liver is the king of metabolism, so it basically has everything. And that's because sometimes you need to convert lipids into glucose as gluconeogenesis. Sometimes you actually have to synthesize these lipids from other things. So yeah, it's really important. And we'll come back to the liver a bit later and give you a whole overview of exactly every single reaction that happens in the liver. So this is a bit more detail into how these lipids are actually broken down from adipose tissue, and then these tags actually go out into the rest of the body, okay? So we have hormones such as adrenaline and glucagon bind to specific receptors. So again, remembering glucagon is released when there's not a lot of glucose, okay? And so in this state, we might want more of these fats being used in our body, okay? So in the cell, tags are broken down into fatty acids and glycerol via lipase, because remember, tags are too big to just go through the membrane. They need to be converted into these other molecules first. Okay, so what happens after they've entered our blood? The glycerol returns to the liver in G3P form, and then it can be fed into other reactions. And the fatty acids, they can bind to serum albumin and can be transported to other tissues. You also have lipoproteins here hanging around as well, transporting them to other tissues as well. Now, these fats can be mobilized to skeletal muscle, the heart, and the renal cortex. And this is pretty important to memorize, but again, we'll come back to that in a second. Then, as it's arrived in the blood, the fatty acids go into the cell, and they dissociate from the albumin, and they enter the cytosol. And the cytosol is where beta oxidation occurs. Beta oxidation occurs in mitochondria. But basically, beta oxidation is just the next part of lipid breakdown, okay? Just a fancy word for saying that tags are converted to acetyl coenzyme A, okay? And then used to produce more energy in the cell, okay? So beta oxidation occurs only in the mitochondria. So that is why red blood cells, they can't undergo beta oxidation because they don't have mitochondria. It's important to realize that it releases 2 acetyl coenzymes A's, and then from there it can enter the citric acid cycle and go into electron transport chain to produce ATP and energy, okay, for its cellular processes. Meanwhile, this produces CO2 and hydrogen atoms, and yeah, you just have to know that. Another really, really important thing to know is that each fatty acid, beta oxidation is repeated 7 times to produce 7 acetylcoenzyme A molecules. Okay, that is really, really important because you might be asked that. Okay, so beta oxidation, don't stress too much about it. It's just going on from the breakdown of lipids, converting these tags into energy. Okay, that's all there is to it. Now, sometimes you might need to actually make fatty acids from other molecules. Okay, so basically just remember that carbohydrate and amino acids can be converted into acetylcoenzyme A's, and these can be converted back into tags. Okay, so this happens if there's an excess amount of glucose sometimes, or too much protein in the body. There's just too much. It doesn't want to store it as glycogen or anything like that, and it converts it into fats. Okay. So anything in excess really does get converted to fat. That's why you get fat. Yeah, moving on to protein metabolism. So you should all know the structure of protein. It's pretty simple. I'm not going to go over that. This is just revision. So first I'm going to go over the digestion and absorption of proteins. So proteins are digested in the stomach by this enzyme called pepsin, which digests collagen. Okay, and that's really important to remember that pepsin actually does digest these proteins. Okay, and then most of these proteins become amino acids, and they're just absorbed in the intestinal lumen, okay, by enterocytes. That's all you have to know about digestion of proteins. Then these proteins are absorbed through the luminal membranes of intestinal epithelial cells in the form of dipeptides, tripeptides, and free amino acids. So all this means is that, again, we can't absorb really large molecules, okay? We want everything broken down into smaller forms, okay? Free amino acids, they're very, very tiny. Dipeptides and tripeptides, also very, very small molecules that we can actually absorb, okay? That's all you really need to know. Just remember, pepsin digests protein, okay? That's most important there. So we have something called the glucose-alanine cycle, which is where we convert protein into energy sources. Now, we don't really want to do this, but sometimes we have to during starvation, okay? Because this gives us another form of acetylcoenzyme A, which can then go into the citric acid cycle to release energy, okay? So this only really happens in starvation. Now, I should have a diagram here. Please memorize this diagram. I know it looks really intense. I promise it's not once you understand what happens. So here, this is happening in starvation. Okay, so you have your muscle protein. In starvation, this muscle protein gets broken down into amino acids. Okay, and it has this amine group. Okay, now we don't like this amine group. We want to get rid of it. Okay, so what happens is that this amine group binds glutamate and gets converted to alpha-ketoglutarate. Okay, and this produces something called alanine. Okay, this alanine from the muscles then goes onto the blood, then finishes up in the liver where it can then be excreted. Okay, so alanine is basically pyruvate being converted to alanine because there's an extra amine group being added on. Okay, so that's the only difference between pyruvate and alanine, is that there's an extra amino group there. So then we want to convert this alanine back into pyruvate, okay, releasing that amine group for it to be excreted in something called the urea cycle, okay? So we have this alpha-ketoglutarate being converted to glutamate, which releases the amine group, okay? And this amine group can then be excreted via the urea cycle as urea, okay? So you just have to remember transamination is glutamate to alpha-ketoglutarate, and deamination is glutamate being releasing the amine group, okay? There's a bit of debate as to whether you actually have to know the urea cycle. I was told last year that we didn't have to memorize it, but apparently some people were told otherwise. I'm not going to be covering it. Honestly, it's a bit too complex for what we're talking about. If you're really keen, go learn it, but I'd recommend against that personally. So if we look back at what's happening here, we have glucose going to pyruvate, okay, and that's transporting our amine to the liver. And then once we've transported our amine group, that pyruvate gets converted back to glucose, and it can go back to our muscle and be stored as glycogen back there. So hopefully that makes a little bit of sense. That's just something you have to know. This goes into a bit more detail about actually what happens to the amino acids. Okay, so we've gone through what happens to that amino group that we don't like and we want to get rid of. So what actually happens to the amino acids is that, oh, sorry, gone to the wrong slide. the amino acids get converted to nucleic acids, amino group, and carbon skeleton, okay? So these nucleic acids are actually just converted back into the body and used later on, okay? Now this carbon skeleton is what we're actually interested in because this goes back into acetyl coenzyme A and then can enter the citric acid cycle. Also important to note here is that sometimes it's indirectly converted into the citric acid cycle because it goes into oxaloacetate. And you should remember that oxaloacetate is one of the intermediate molecules in the citric acid cycle. So either goes straight into the citric acid cycle via oxaloacetate or first goes into acetyl coenzyme A and then goes into the citric acid cycle. That's all you really have to know about amino acids. So we'll keep going. So ketones are a little bit random. All you have to know is that they're a chemical substance that the body makes when it doesn't have enough insulin in the blood, okay? Or it doesn't have enough glucose in the blood, okay? Because remember, because that's when you wouldn't have insulin in the body. And they're formed from acetyl coenzyme A in the liver during oxidation of fatty acids, okay? That's all you have to know about that. And just know that high ketone levels can occur in severe starvation or in diabetes. So if you see these two words in your exam, just think about the fact that there could be a lot of ketones made. So moving on to alcohol metabolism. So alcohol metabolism, so alcohol you should know is the same thing as ethanol, okay? In our bodies, it gets converted to acetaldehyde. Acetaldehyde gets converted to acetate. This acetate gets converted to acetyl coenzyme A. And then this acetyl-CoA specifically gets converted into fatty acids or ketone bodies. Now that's why you can think of fatty liver or, you know, beer bellies, I think they're called, yeah. Because if you drink too much ethanol, you actually get too many fatty acids being produced. I think we also had a question. Oh, yes, okay. I will go back to the previous slide, sorry. Is it this one that you'd like me to go back to, Savannah? Okay, sorry, okay, sorry, just someone wants to have a bit of a look at the slide. Yeah, honestly, the only thing you have to know about ketones is that they occur in severe starvation or in diabetes. I also have another question. So is there anything we don't have to memorize from the last slide, the urea cycle? Yes, so you don't really have to know the urea cycle. There's a bit of debate and some people will tell you to memorize it. I got told by my tutor we didn't have to, but yeah, it's a bit complicated. So I'm going to move on to alcohol metabolism. Hopefully that was enough time. Okay, so back to alcohol metabolism. So that's why if you drink a lot of alcohol, you get a lot of fatty acids produced and a lot of fat in the body, okay? This is some diagram here, which just shows you what happens. Now it's really important to realize that NAD plus gets converted to NADH here and here as well, okay? So they'll come into context a little bit later, okay? And remember in the cellular respiration presentation, I mentioned that if you have a lot of alcohol, you're producing a lot of NADH, and that feeds back into that anaerobic link reaction, producing a lot of lactate. And we'll cover that a bit more in a second. So acetaldehyde, this molecule here, is actually really poisonous, and your body really wants to get rid of it, okay? So some people actually have acetaldehyde dehydrogenase deficiency, which leads up to a bit of acetaldehyde. Sorry, going back to the last slide, I forgot to mention, is that these reactions here, they're powered by enzymes, okay? So to convert ethanol to acetaldehyde, you have something called alcohol dehydrogenase, okay? And that powers this reaction. To power the acetaldehyde to acetate, you have this enzyme called acetaldehyde dehydrogenase, which really powers this reaction here. If you have a deficiency in any of these, it's going to make it a lot harder to continue this reaction, which is why some people have a deficiency of acetaldehyde dehydrogenase. Sorry, that is so hard to pronounce, which leads to a buildup of acetaldehyde, which is poisonous. And this causes face flushing, vomiting, and nausea. So now we have, I'm going to explain the buildup of NADH and its effect on metabolism. So as you should probably have already know, I've said this a billion times, but NAD plus gets converted to NADH, okay? And this causes a buildup of NADH. And if you go back to the anaerobic link reaction, that powers the conversion of pyruvate to lactate, causing a lot of lactate being built up, okay? It's also, if you have a lot of NAD plus being used up, okay, if you look at the citric acid cycle, which I have here, if you don't have a lot of NAD plus left in the body, you're not going to be able to power this citric acid cycle, okay? Because there's a lot of this NAD plus being used up, okay? Yeah, so on the next slide, we go into a bit more detail about that. An excess of NADH can cause a buildup of lactic acid in the body, and this is called lactic acidosis, which lowers the overall pH of the body. And remember, we treat that with sodium bicarbonate to lift that pH level back to normal. And you might actually be doing this one day as doctors, so that's finally something exciting that's clinically relevant. This also, as I mentioned, reduced the rate of Krebs cycle, or citric acid cycle, whatever you prefer to call it, which results in a lower level of ATP output, which is why people with alcohol abuse problems can feel really fatigued, because they actually lack ATP and lack energy. And lactic acidosis also causes fatigue as well, so it's sort of a double whammy. So I'm just going to move into a bit of a summary of the liver overall and its metabolic function. So remember, liver is king, and it's basically where most of the metabolism occurs, okay? So remember, the liver stores large amounts of glycogen and converts other molecules to glucose, and acts as a primary site for gluconeogenesis. Okay, so that's really important to remember for carbohydrates. For fat metabolism, remember, it can convert lipids to acetyl coenzyme A via beta oxidation. It's also the place of synthesis of large amounts of cholesterol, phospholipids, and lipoproteins. And almost all fat synthesis from carbohydrates and proteins occurs in the liver. It can occur in other places, but liver is the primary spot for it. And then for protein metabolism, remember, the liver is the place of deamination of amino acids, so getting rid of that amino group in the form of urea, okay? It can also form plasma proteins as well. That's something you don't actually really need to know, but fun fact. So next we have a bit of an overview of different energy sources for different tissues. So here, the heart, this is a table that you really do need to memorize, like ASAP. I can't tell you any other way, you just have to memorize this, okay? So during, just in your normal state, when you're not starving, okay, you're pretty well fed, your heart primarily runs on lipids, okay? Your brain uses glucose, as do your red blood cells, and your skeletal muscles use lipids, okay? Now, when you're starving, the heart uses both glucose and ketones. The brain uses ketones, red blood cells use glucose, and skeletal muscles use protein. So here it's important to note is that the heart, remember, the heart can't undergo anaerobic respiration, okay? So if the heart runs out of sources of energy, it literally starts to die, and then if the heart dies, we die. So you cannot have that happen. That's why the heart is quite versatile and uses pretty much all these different forms of lipids, glucose, and ketones. Okay, that's really, really important to remember. They can use all these ones. Now the brain is a bit more specific. Brain uses glucose, okay, that's the main one, but it can also convert to ketones. Okay, I'll go into a bit more detail about the brain in a second. Now remember, red blood cells, they don't have any organelles, they don't have mitochondria, so they can only undergo anaerobic respiration. So they have to use glucose, okay? That's pretty easy to remember. And this is basically the reason why we always have to have some sort of glucose in our body, okay? For those red blood cells to use them all the time. Now, skeletal muscle, they mainly use lipids as a normal energy source, but in starvation, as we already saw, they might have to use proteins, which is not good because we'd rather have our proteins in our muscle, okay? So this is basically a bit more detailed overview of this. And yeah, it basically says exactly what I just said. Now, the reason why the heart prefers to use lipids is because lipids are actually more energy dense. Okay, they release a lot more energy than glucose, all right? that's why it's a preferred source, because there's just more energy coming from lipids. Now, the brain prefers blood glucose, but can use ketones. Like the heart, it doesn't store energy, so you need a constant supply from other sources. Now, the brain requires a lot of energy, and it counts for about 20% of oxygen use, okay? So it's working all the time, okay? And you always need a lot of energy for the brain. Now, the reason why, I had a research into it, and I think some of you might be about to ask the question as to why the brain can't use lipids. And the reason for that is actually very, very complex. But basically, lipid beta oxidation can actually use up a lot of energy in creating it and use up a lot of oxygen. And the brain is already using about 20% of our oxygen normally. And if you're requiring even more oxygen, it's not a good situation. And also technically, in lipid metabolism, there's sometimes these poisonous intermediate molecules that are made, and the brain can't handle that. So yeah, that's why the brain does not like using lipids. But that's advanced knowledge that you don't really have to know. That's just a fun fact. Oh, I have a question. Okay, what is the question? Okay, so yeah, this lecture will probably happen in another 10 minutes. Yeah, another 10 minutes for this lecture. Okay, red blood cells. Okay, so red blood cells only use glucose, and you already know that because they don't have mitochondria. Yeah, there's nothing really else to say. Oh, just don't forget the pentose phosphate pathway, which I talked about in our last lecture. It's just an alternative to glycolysis, which can also be used. And then skeletal muscle energy use. So this is quite important to memorize and to understand, because you very well might have a question on this, okay? So in the well-fed state, as you know, skeletal muscles mainly sources energy from lipids, okay? At a moderately active level, they use glucose found from glycogen, okay? And remember, unlike the liver, it's glucose 6-phosphate is used directly, okay? At an extremely active level, it undergoes anaerobic respiration, and basically the glycogen becomes lactate, okay? That's all you really have to know there. In short bursts, this is really important, I haven't actually mentioned this before. In short bursts, skeletal muscle uses creatine phosphate, which phosphorylates ADP to ATP in one very quick step, okay? And creatine kinase is the enzyme used to power this reaction. This is really important to remember because you very well might have a question on this. And the reason for why it uses creatine phosphate is because it's so quick to phosphorylate in just one step, ADP to ATP. So yeah, it's really just a quick reaction. And when you need a lot of energy really quickly, it just comes from this. During starvation, they break down the proteins into amino acids, and that can be converted to glucose for use in other tissues. Now we're going to have some questions. So I know some of you might be wanting to cover great metabolic race, but I really don't want to give you the answers to those questions before you've done it. And I don't think faculty would be very happy with me giving out answers before an active learning. So we're going to do this quiz. Oh, I have another chat. Okay. So a patient has a blood test revealing that they have a high cholesterol. Which of the following substances is elevated? So the answer is B, correct. Joshua, you are correct. Very good. So just remembering that LDL is bad cholesterol because it transports all this cholesterol to the other tissues. And HDL is the good cholesterol that eats up all that cholesterol. Okay, next question. Fatty acids bind to what substance when being transported in the blood? Anyone remember? Yes, very good. B, great. Albumin. That's really important to remember. Okay, that was a pretty easy question. Oh, no, I gave you the answer. Pretend you didn't see that answer. For each fatty acid undergoing beta oxidation, how many acetylcoenzyme A's are produced? Okay, yes, it's C. Very good. Yep, that's a bit sad you saw the answer, but that's okay. Question 4. Which reaction does the enzyme acetaldehyde dehydrogenase power? Yeah, very good. So this one can be a bit confusing with the other enzyme. C just has acetate and aldehyde in its name. So that's why it's this one. Okay, we just have a question. For question 3, doesn't 16 carbon fatty acids produce 8 acetylcoenzyme 8s? So that's a very good question. But in beta oxidation, you just have to remember that carbon dioxide is also produced. Okay, so there's always going to be one molecule of CO2 released. That's why even though it's 16 carbons, it gets broken down into 7 and 7. Okay, and there's 2 carbon dioxides produced as well. Okay, so that's why it's not 8 acetylcoenzyme. Yeah, just remember that. Okay, question 5. Excessive alcohol abuse can cause rash, lactic acidosis, fatigue, hair loss. Oh, very good. Someone picked up on my trick question. It is both B and C. Very good. Yes. It's actually really hard to come up with other options for this because like excessive alcohol abuse can cause a lot of problems. So don't do that. Question number 6. Alfie the dog has to go on a diet and has not had food for the last 24 hours. Poor thing. What process is happening in his body? Have a think. Death. No. Okay. Yes, the answer is A. Gluconeogenesis. Correct. Okay. Question 7. At the very start of a 100-meter puppy race, Alfie is using what energy source? Yes, so in this, this is actually a bit of a poorly worded question. It's actually C. So if it's a very intense sort of burst of energy at the beginning, it will be creatine phosphate because remember, it's really quick to phosphorylate ADP to ATP in one step. Technically, it can be glucose if it's just leisurely running, but this is really intense at the beginning. Sorry, that was my bad, poorly worded question. Otherwise, you're correct. So I think that's pretty much it for this metabolism lecture. I will be covering Great Metabolic Race. Please tell me if you prefer that as a concept video or as an actual presentation. You can just message me that. And on these slides, you can see that we've had a lot of med law and ethics uploaded as well to HCAS on our Google Drive. So take some time to have a look through that. Send us any questions. And if you'd like a presentation, please tell us. We really, we're here to help you, okay? And we really want some feedback on what you guys find helpful and not as helpful. So that concludes this presentation for me. Hope you guys all have a lovely day. If you have any questions, please feel free to message any of us. We're more than happy to talk to someone. Otherwise it gets lonely. So yeah, hopefully that was kind of helpful. Yeah, so see you around. Oh yes, fill in our Google form if you like it instead. So yeah, thank you very much.